Hi everybody, Mrs. Singerly here, and today we're going to continue with our biochemistry unit, and we're going to start learning about carbohydrates. Specifically, we're going to be talking about monosaccharides, disaccharides, and we're going to talk a little bit about polysaccharides, okay? So carbohydrates. Carbohydrates are organic compounds, right? That means they have carbon and hydrogen, and we have to know what elements they contain. So they contain the elements carbon, hydrogen, and oxygen. I remember it as CHO. So those are the three elements that are in the carbohydrates. And our carbohydrates are always going to have a ratio of hydrogen to oxygen atoms that is always 2 to 1. So that means that there's double the amount of hydrogens than there is oxygen in the carbohydrates. And we can kind of always remember, like with carbohydrates, the carbos for carbon, and the hydrates, like our hydroxyl group, right, has that hydrogen and oxygen. And that hydrogen to oxygen ratio is always going to be 2 to 1. Now, carbohydrates are broken down to provide cells with immediate energy. So that's one of their primary functions, is that they're an energy source. We can use them for an immediate energy source, or we can store carbohydrates. We're using them to store energy. We can also combine them into polymers or convert them into other organic molecules as well. And there's different kinds of carbohydrates. There's starchy carbohydrates that you're probably familiar with, things like bread, pasta, and rice. But then there's also sweet tasting carbohydrates like candy and honey and syrup. So we're going to start with the simplest kind of sugar, a monosaccharide. So mono means one, right? And if we were to go outside and talk mano a mano, we're going to be going outside and talking one-on-one. So mono means one, and saccharide or saccharin refers to something sweet. And so monosaccharides really mean just one sugar. And foods that contain monosaccharides are going to also taste very sweet. So these are going to be things like the candy and the honey and the syrup. Monosaccharides are going to be monomers, right? They're going to make our more complex carbohydrates, which are going to be polymers. So we're going to be stringing these monosaccharides, these monomers together to make our complex polymer. And the most common example you're going to see of a monosaccharide is glucose. And we're going to also notice that when we're talking about sugars, the names for most of them end in OSE. So things like glucose and galactose, fructose, sucrose, lactose, those are all types of sugars, and they all have that OSE ending. Now, what's really interesting about glucose, galactose, and fructose is there's something that we call or refer to as an isomer. An isomer means that they have the same molecular formula, which we're going to learn as C6H12O6, but they have slightly different structures. So let's look at the structure of glucose. A structure of glucose, it's going to look like a hexagon, so it's going to kind of look like a stop sign. And what you're seeing on the screen is its structural formula. Now, this question asks, what functional groups are present in a glucose molecule? And the answer is a hydroxyl group. So if you'll notice, right, we've got a hydroxyl group here, we've got a hydroxyl group here, hydroxyl group here. And so these are kind of the functional groups. And we're paying attention to those because those are going to be where bonding occurs. Now, if this is our structural formula, so what it looks like, the molecular formula is going to be how many atoms of each we have. And so in a molecule of glucose, we have 6 carbons, 12 hydrogens, and 6 oxygens. And you'll notice that we have 12 hydrogens to 6 oxygens. And if you think about that, if it was in a fraction, 12 over 6. That would reduce down to 2/1, and that's our ratio. There's double the amount of hydrogens as there is to oxygens. And so that's our structure of glucose, our monosaccharide. Now, monosaccharides can be classified based on the number of carbon atoms in each molecule. And so there are other types of monosaccharides. There are trioses, which only have 3 carbons in them. There's pentoses that have 5 carbons. That's going to be like ribose and deoxyribose. Those are the sugars that we're going to find in molecules like DNA and RNA. I know my face is kind of over the picture over here, but this is my pentose sugar. It kind of looks like a house if you look at just this inner ring structure. And then you have hexoses, and those are going to be things like glucose, galactose, and fructose. And you can really see that, say, with glucose and galactose, they have this hexagon stop sign looking structure to them, and that's going to help us recognize it. And so glucose, right, is it a triose, a pentose, or a hexose? Remember, its molecular formula is C6, so it's going to be a hexose. Now, the shape of monosaccharides, technically monosaccharides can be linear or they can form rings. Now, most of the time on the AICE exam, you're going to see pictures of them in this ring shape. But I did want you to know that they can be linear, but as we have more carbons in, say, like our pentose and hexose sugars, they're long enough to close up on itself and form this kind of more stable ring structure. And a lot of times when monosaccharides are in liquids, like in water, they're more apt to form this ring structure, and we know that our body is made up of a lot of water, and so when these molecules get put in water, that's usually the shape that they're going to be in. Now, sometimes you'll see it as this abbreviated shape. Notice that where all these kind of points are, those are where the carbons are, but they've kind of omitted putting the carbons in. So it's sometimes where you see these, corners. If the carbons aren't listed there, they are there. They're just like abbreviated in the picture. Okay? But most of the time, like for our class, when I show you pictures, I'll try to include these carbons in there. There are different types of glucose molecules. So our glucose, our main monosaccharide, which is C6, H12, O6, comes in 2 different forms, an alpha form and a beta form. And you may have never seen these Greek letters before, but this is the symbol for alpha, and this is the symbol for beta. And so this is an alpha glucose and a beta glucose. And how we can tell the 2 of them apart is where the hydroxyls are. Is the hydroxyl group above or below the plane of the ring? Now, why this is going to be important is that when we talk about building polysaccharides, it's going to matter whether or not we're making our polymer with alpha glucose molecules or beta glucose molecules. And the polymers that we make, depending on the type of glucose molecule, can change its function. So our alpha glucose, the hydroxyl group, is below the plane of the ring. With a beta glucose molecule, our hydroxyl group is above the plane of the ring. And it's difficult to remember because it's the opposite of what you think. So alpha is down and beta is up. Okay, so alpha is down, beta is up, and that's something that you will have to practice with like our Quizlet cards, trying to remember that the alpha has the hydroxyl group down and the beta has the hydroxyl group up. Thumbs up, thumbs down, okay? Now, how do we take those monosaccharides and make more complex carbohydrates? Right? Remember, we're doing this through the process of synthesis, which in AICE we call, condensation reactions. And these condensation reactions is the process by which we're taking smaller molecules and we're combining them to make much larger molecules. And the smaller molecules that we're using to combine to make larger, more complex carbohydrates are going to be those monosaccharides that we talked about, things like glucose and galactose and fructose. And because we're using them to build things that are more complex, right, we would say that the monosaccharides are the monomers of these complex carbohydrates that we're going to build. So first thing that we're going to build is what we call a disaccharide, right? The prefix di means 2, saccharide means sugar. So these are double sugars. And this is what we'd call a disaccharide. And one example that you need to know is maltose. Maltose is an example of a disaccharide, and here is a picture of it below. Notice we've got 2 sugars that are bonded together, that are joined together. And that's all a disaccharide is 2 monosaccharides covalently bonded together. And our molecular formula is going to be C12H22O11. And you'll notice that I know that this is a carbohydrate because my hydrogen to oxygen ratio is 2:1. Now you might be thinking to yourself, wait a second, Mrs. Singerly, if I'm taking two glucose molecules and putting them together, that's C6H12O6 plus C6H12O6. Why isn't it C12H24O12? We're missing some atoms here. What happened? That has to do with our condensation reaction that I'm going to show you next. Because in order to bond those two molecules, remember, in a condensation reaction, I have to remove water. And so I have to kind of remove those atoms. It's still going to be a product. It's technically going to be called a byproduct, right? Something that we made that we didn't intend to make. But it's not going to be in my maltose molecule or my disaccharide molecule. So let's take a look at exactly what happens. I got the little water dripping there. I don't know if you can hear it. That's the water being taken out. So here is my two glucose molecules. And you'll notice here are my hydroxyl groups. You'll notice that these two are lined up side by side. And my two hydrogens are going to combine with my one oxygen. So I'm going to have this hydrogen from here and my hydrogen from here and this oxygen. They're going to combine to form water. Okay, so there's my water that came out. And it's going to form kind of this oxygen bridge between the two, so that way they can be bonded together. And that bond, right, is a covalent bond, and they named these. So all the bonds that are going to hold together our polymers are going to have special names. And so the name for what the type of covalent bond that holds our two monosaccharides together is going to be called a glycosidic bond. Sometimes it's also referred to as a glycosidic linkage. Now I want you to remember, right, in chemical reactions, our reactants are on the left, our products are on the right. So my reactants in the condensation reaction of a disaccharide, specifically making maltose, I'm taking glucose and glucose, and I'm putting them together to make maltose. So glucose and glucose are my reactants. Maltose is my product. And then because I had to take out water, that's also showing up here on the product side, right? It's a byproduct, something that I made that I didn't intend to make. And so I want you to remember with chemical reactions that matter isn't being lost or added. All we're doing is rearranging some of the atoms. So all we're doing is rearranging it a bit. So we're taking water out and making that its own separate molecule. And when we did that, it formed a glycosidic bond to make my disaccharide. There's my water. Now, the opposite of a condensation reaction would be hydrolysis. So if you were to eat a food product that had maltose in it, your body's going to digest it and break it down by the process of hydrolysis. And it's just the reverse reaction. And this time I'm adding water to break that glycosidic linkage, and I'm breaking it apart back into my two individual glucose molecules. And you can see where this water is added. An oxygen and a hydrogen went to one of the glucose molecules, and the last hydrogen went to this glucose molecule, and that separated the two of them. So if we were to go through this and say, okay, what are my reactants, right? So I'm looking for everything to the left of the arrow. That would have been maltose and water. And that when I added the water to my maltose, what happened? It broke apart my two glucose molecules, and I end up with glucose plus glucose. Now, I know you're asking yourself, but Mrs. Singerly, are there more molecules? Yes, there are. There's more than just maltose in our disaccharides. And we're going to be doing a testing for nutrients lab in another week where you'll get to see kind of how we test for these different disaccharides. And so examples, right? Maltose, we already know. Maltose is a disaccharide that's made out of glucose and glucose. So here's our maltose molecule, and it's made out of two glucose molecules. Something like sucrose. Sucrose is you know, the sugar that you'd find in the shop, like the sugar that you'd find at the store. And it's made out of, it's a disaccharide that's made out of glucose and fructose put together. So I remember that because fructose is a sugar that's found in fruit. And so if you think about, okay, like sucrose is sweet and like fruit is sweet. And so I kind of remember that fructose is the glucose and fructose put together. And then lactose is made out of a disaccharide that's made out of glucose and galactose. And lactose is actually the sugar that's found in milk. And if you're lactose intolerant, you can't digest the sugar that's found in milk. And that's what gives you the stomach upset, the vomiting, the diarrhea, and all the bad stuff. Now, we can keep this process going, right? We just took 2 monosaccharides and put them together to make a disaccharide. But I can keep this I can add a 3rd one, a 4th one, a 5th one, a 6th one, on and on and on and on. This process of synthesis or condensation reactions can continue repeatedly until we get a chain of many monosaccharides. And that's all formed by bonding all of those monosaccharides together through covalent bonds, specific covalent bonds called glycosidic bonds, or those glycosidic linkages. And remember that every time I put 2 monosaccharides together, every time I do that, I have to remove a water molecule, right? And so that's why my maltose molecular formula was C12H22O11. Right, and not 24O12, because I had to subtract out a water to stick those 2 together. And so every time I'm adding on a monosaccharide, I'm removing water and forming a glycosidic bond. And since polysaccharides are chains of many monosaccharides connected by these glycosidic linkages, they are known as polymers. Right, so polysaccharides is going to, poly means many, so many sugars. And they're going to be our polymer for our carbohydrate macromolecule category. And so our monosaccharides then are therefore going to be our monomers. And what you see on the bottom of your screen is a really oversimplified picture of putting these monosaccharides together to make a polysaccharide. Now, the final molecule is not going to be just 4 monosaccharides linked together. They can be thousands of monosaccharides long, forming this giant macromolecule. And some important polysaccharides we're going to talk about in the next video that you guys are going to watch tomorrow is going to be on starch, which is a really common polysaccharide, glycogen, cellulose, and chitin. That's what we're going to be learning about tomorrow. And they're all polymers of glucose, but they function differently because of the different forms, right? Alpha, or sorry, I did that backwards. Beta. Oh my goodness, I can't. What am I doing, Mrs. Singerly? 2 different forms of glucose. Alpha, which has the hydroxyl group pointing down, and beta, which has the hydroxyl group pointing up. And that's going to give these different functions, which are going to be really important. And these polysaccharides that we're going to be talking about, right, like starch, like, you know, like potato, pasta, they don't taste sweet. They're not sugars. So that's what we're going to be talking about in our next video. And that is it for today. I hope you found this video helpful. And remember, if you have questions, write them down, email, private comments in Google Classroom. Let me know, and then I'll come back later in the week and we'll kind of go over all of those questions, okay? Have a great rest of your day, and I hope this was helpful.